Now, of course, microbiology did not start in the genomic era. And I like to go back to the beginning of any story. I think it's a really useful place to start and to ground ourselves and contextualize. So this is actually the tomb of Antony van Leeuwenhoek in Delft in the Netherlands. He was really the first microbiologist. He was the first one to make observations of these microscopic animalcules and describe them formally. So when we're talking about microbiology, what are microbes? Well, the topic of this course is bacteria and fungi, but we also have parasites and viruses, which will be covered in other courses in the DVM curriculum, or for those of you on YouTube, you will have to find that content elsewhere. So how do we differentiate bacteria from other organisms? What are some features of prokaryotes that allow us to distinguish them from eukaryotes, from us, or from eukaryotic microorganisms like yeast, protozoa, and so on? Size is one feature that we can use. Bacteria, generally speaking, are bigger than viruses and smaller than eukaryotic cells. They're single-celled organisms. They possess a cell wall composed of peptidoglycan. They reproduce asexually through binary fission. They're capable of lateral or horizontal gene transfer. They have a short generation time. They typically, although not always, have a circular chromosome. Unlike eukaryotes, they lack a nucleus and other membrane-bound organelles. And prokaryotes have unique metabolic requirements and unique abilities. So how do we tell prokaryotes apart from each other? How do we differentiate one species from another? Well, when we talk about differentiating species, this is really the field of taxonomy. So how we group things on the basis of shared characteristics. And this also goes back to the historical system developed by Carl Linnaeus, Linnaean classification or binomial nomenclature, where we refer to an organism by its genus and species. So if we look at the common bacteria, Staph aureus, Staphylococcus aureus, you can see here's its full taxonomic designation. Our genus and species are always italicized. And then we have our higher taxonomic orders, family, order, class, phylum, kingdom, and domain. Throughout this course, I'm primarily going to be talking at the level of the genus and species, but we will occasionally refer to bacterial families or bacterial orders. Bacterio.net is a really useful resource for getting up-to-date information on bacterial classification. Microbiologists love to reclassify organisms, to split species apart or lump things together. So this is a place where you can find the most up-to-date recognized list of bacterial names. Now, entering the medical sciences really requires you to learn another language. There's, of course, a lot of physiology, anatomy, pathology that you need to learn, but all of this is underpinned by essentially a new vocabulary. And in microbiology, that's no different. In my opinion, it's very important to know the full name of a microbe. So I expect my students to be able to know that E. coli is actually Escherichia coli. And the reason that that's important is that we need to be able to speak precisely, and abbreviations can be quite misleading. So if we were talking about M. bovis, for instance, this could refer to Moraxella bovis, Mycoplasma bovis, or Mycobacterium bovis, three different bacterial species with dramatically different implications for the health of an individual animal, the herd, and even regulatory implications. On the other hand, if we look at multiple genera with the same species name, we have a pyogenes in both in Streptococcus, Truparella, Bacteroides, and Corynebacterium. So knowing the full name of the organism is very, very important. If we think about taxonomy in terms of macroorganisms, I think it's a little bit easier to understand. So we have A vertebral column, yes or no? Is there a hinged jaw? Do we have legs? Do we have an egg with an amnion? Do we have hair? These are all observable characteristics. But how do we differentiate bacteria from each other when they're microscopic? What types of features of these organisms can we use in order to say we have one species as opposed to another? The classical description of bacterial species relies on a number of characteristics. Maybe the most famous and one of the most basic and useful is the gram stain characteristic. This is based on the structure of the cell wall. So on the left here, you can see a cartoon of the cell wall of gram-positive bacteria. And on the right, you can see how a gram-negative bacteria would look. There's a few key differences between these organisms. So most notably, this very thick peptidoglycan wall in gram-positives, quite a bit thinner in gram-negatives, and then the presence of an outer membrane surrounding gram-negative bacteria. I'll put a link to a video where the procedure for performing a gram stain is described up here. Gram staining is a 4-step process. First, slides are treated with crystal violet, which is our primary stain, followed by grams iodine. The third step is to decolorize the slide, so we use acetone, which is able to wash away the crystal violet and iodine complexes of gram-negative bacteria. That thin layer of peptidoglycan doesn't hold on to the stain as well. We then follow up our acetone with carbofuscin, a counter stain that's pink color, and that allows us to visualize our gram-negative bacteria. Here's an example. On the left, we have the gram-positive Streptococcus agalactia. You can see these nice purple cocci. And then on the right, we have our gram-negative Pasteurella multocida, a small gram-negative rod. We can also describe our bacteria based on their cellular morphology, whether they're coccoid or round, rod-shaped, or spirochetes or helically shaped. Atmospheric requirements are another really useful and diagnostically important characteristic, whether the organism grows in ambient air, requires anaerobic conditions, microaerophilic conditions, or CO2 enrichment or capnophilic conditions. A variety of biochemical characteristics or really metabolic requirements. Oftentimes, these are colorimetric assays that are based on the ability of an organism to utilize a particular substrate or produce acid from a particular substrate. We can also look at hemolytic patterns of the bacteria. So by growing an organism on sheep's blood agar, we can look for an interaction between the bacteria and the erythrocytes or red blood cells in the media. On the left, we have an alpha hemolytic bacteria. Alpha hemolysis refers to partial hemolysis or the presence of methemoglobin within those erythrocytes. In the center, we have beta hemolysis, which is complete hemolysis or breakdown of those erythrocytes in the media, leading to clearing zones surrounding the bacterial colonies. And then on the right, we have gamma hemolysis, which is a little bit of a misnomer because what we're actually referring to is no hemolysis at all. We're now in the era of molecular microbiology, and so it's no surprise that molecular techniques have become increasingly important in the identification of bacteria. Classical taxonomic approaches do have limitations. Maintaining a wide inventory of biochemical tests can be costly and tedious, and these tests are quite time-consuming to do. So whether we're using PCR and sequencing of amplicons or whole genome sequencing, this has become increasingly important in both the diagnostic lab and in research applications. But how do we use DNA sequences in order to identify a species? And what actually constitutes a level of similarity that indicates we have the same species? What you can see here is a phylogenetic tree of the CPN60 universal target for a variety of staphylococcal species. So this is a housekeeping gene. It's something that's well conserved, and it's a target that we can compare the sequence between species or between isolates to determine how closely related they are to each other. When looking at a phylogenetic tree laid out in this way, what we're concerned with are the horizontal distances between samples. So for instance, these two isolates here, aureus APR25 and aureus ATCC 13565, there's no horizontal distance, which indicates that these two sequences are actually identical to each other. If we were to compare these aureus to Staph hemolyticus here, you can see there's a wide horizontal distance, which indicates that there's divergence of these sequences. Is there a cutoff we can apply where we can differentiate between species? So for instance, this red line here, any branch points to the right indicate intraspecific diversity, whereas branch points to the left differentiate one species from another. On the right here, we have a phylogenetic tree, again, of CPN60. This is only Staph aureus. So you can see that not only do we have differences between species, but there's also variability within recognized species. Comparing organisms to each other on the basis of phylogenetic analysis might initially sound quite simple, but it can actually be fairly challenging. Here we have a phylogenetic tree, again, of the CPN60 universal target of Escherichia coli and Shigella species. Now, these are actually separate genera, but they're very, very closely related to each other. You can see here we have Shigella and E. coli co-clustering, so they actually have identical CPN60 sequences, even though they're different at the genus level. So the molecular description of a species is perhaps not as simple as it initially appears, and is actually quite an active area of research in both microbiology and taxonomic studies of higher organisms as well. Widespread whole genome sequencing is giving us really great new insights into really the tree of life, and certainly keeping microbiology exciting. Throughout this course, I'm going to include some new terms at the end of each lecture to ensure that you have the vocabulary required to understand the content. So today we have prokaryote and eukaryote. Finally, on the last slide, I include some reflection questions for self-assessment. I hope that these questions will serve as a study guide for you to reflect back on the content of each lecture and ensure that you understand the key points.